Welcome to episode 41 of the Breaking Bio podcast. I'm Morgan Jackson, PhD student at the University of Guelph in Ontario, Canada. We got a new and exciting guest, like always, but first we'll go to our regular hosts.
Hi, my name is Heidi. I'm a postdoc at the University of Texas at Austin.
Hi, I'm Stephen Hamlin, postdoc at the University of New South Wales in Sydney, Australia.
Hi, I'm Tom Housley. I'm a PhD student at the University of Stirling in Scotland.
And as I mentioned, our guest today is Michael Whitehead, who's a postdoc who spends half of his time at the Australian National University and the other half of his time at KwaZulu-Natal University in South Africa. So welcome to the podcast, Michael. What exactly do you do when you're continent hopping?
Thanks, guys. Right now I'm studying pollination projects and plant mating. So when I'm in Australia, Australia side of things, I study orchids and their interactions with ectomycorrhizal fungi. And in South Africa, I'm studying long-tongued fly pollination, as well as some sexually deceptive orchids.
So it's going to be pretty busy to be doing two projects in alternating seasons. I guess you could double your field time, I guess, but we won't make you choose which one's your favorite. I suppose that's what we mean.
Well, I'm still learning how it goes. I mean, it's all good in theory because I do get two field seasons back to back, but I've only done it once, and it's working so far. But I think I'll be glad to settle down at the end of the next trip because the next trip's probably going to be my last trip to South Africa for a little while. It doesn't make relationships or finding the time for lab work and writing that easy, but I do get a lot of field time, which is good fun.
So where in South Africa have you been doing your research?
So my research has been in the Drakensberg mountain range, which is just inland from Durban. It's these beautiful summer rainfall alpine meadows, and for a botanist, it's been pretty inspiring to go there and see the amazing diversity of flowering plants they have, and especially everything being below your knees. It's a really nice place to work for a field biologist. I can see why so much work has got done there because the scenery is just spectacular as well. So I was there last summer on a behavioural project looking at long-tongued flies, which are these nemestrinid. There's Morgan. He's happy we're onto flies already.
Exactly.
They're a major part of the pollinator fauna in this really diverse community of flowering plants. So there's been a whole guild of flowers that have evolved to use the long-tongued flies as their pollen vectors. And the flies have evolved, proboscis is up to around 6 or 7, and I think the largest species is around 8 centimeter proboscis to get to the bottom of long tube nectar flowers. And the colors of the plants that they pollinate, they're fairly restricted to pink, white, and blue. So I was there to find out how much of that phenomenon of convergence on those colors is due to pollinators learning and being conditioned onto those colors, as opposed to perhaps a sensory bias that the animals might have innately.
Do they show a preference for a certain color over the other ones? Like, is pink their favorite, and then white?
So I tested preferences in the field by making little fake flowers and then painting them. They're made out of acetate overhead transparencies, and I presented it on our highly scientific choice stick, which is just a little T-bar with a pink flower on one side and a white flower on the other side. Well, they're inflorescences, and then by presenting that to a foraging pollinator, I was giving it a binary choice. And I found that in communities where the flies were feeding strictly on pink flowers, that they showed a pink preference, and they ignored the white relatively. And then in communities where the flies could only feed on white flowers, then they had a white preference. And then in a mixed community where they were feeding on white, blue, and pink, then there was no clear preference between those things. So they obviously are able to, well, that still doesn't answer the question of whether it's conditioning or an innate sensory bias that might be local to different populations, but there's definitely a range of preferences they can display.
How far apart are populations?
They're probably around 30 or 40 Ks. So we're looking at populations that could potentially not have gene flow between them, and so the only way to really answer this question now, which is what I'll be trying to do next season, is to either establish that you have gene flow between two populations that have different preferences, in which case you must be looking at learning, or to capture flies, put them in a cage, and then try to switch a preference by giving them flowers that they might not initially have been conditioned on. You can give them a nectar reward in order to try to switch the preference. So that's what I'm going to try next season. I tried that last season, but they all died, basically.
Tom knows that story.
Wow.
There's basically nothing known about these flies, so they're a challenge to work on from that perspective. I'd love to be able to rear a lab population, but that's not going to happen anytime soon.
It'd be tough. We should just point out, though, so you said that some of their tongues were, 8 centimeters long. The flies are 1-2 centimeters long. Their tongues are vastly just misproportioned for their bodies.
Yeah.
So they're pretty cool flies, for sure.
My supervisor calls them stately creatures. Yeah.
Awesome.
They are one of the more charismatic of South Africa's fauna. You can forget about cheetahs and leopards and everything. These guys are pretty special.
I agree. That's the only reason people should be going for Africa is to go look at the flies. So.
Yeah, a lot of people said instead of like the big 5 for Africa, they want to call it the big 6 and include the flies.
Well, I started a lab poll when I was there about what the pollinator big 5 might be. Because thinking that it was such a good marketing tool for South African tourism, having the regular big 5, then might be a good way to a good bit of outreach to put together a pollinator big 5. And there was a lot of indecision in the pollinator lab about who might be the big 5, and it turned into about the big 20, as it does when you talk to scientists and they have to split hairs all the way along.
Awesome. So these orchids that you're working on, just doing a quick scan of your research, it looks like some of these orchids are actually giving nectar, and some of these orchids, or flowers rather, are not giving nectar to these flies. And so, some of these flowers are almost acting like parasites, getting the pollination for free without paying the flies off. Is this a common strategy in the area, or is this one or two creeping under?
For orchids, it's a very common strategy, and an orchid will only ever it'll only ever exist in a community in a fairly a rewardless orchid usually exists in fairly low frequencies, and so you have three main strategies of orchid deception, and they are Batesian mimicry, or Batesian food mimicry, where they model themselves on a particular they model themselves on a particular nectar-bearing plant, and we have cases of that in South Africa. And then there is what's called generalized food deception. So they look just generally like a nectar-bearing flower while not resembling any one specific species. And then there's sexual deception, which is the mimicry of insect mating signals in order to deceive insects into mating with the flower. And I think it's something like a third of all orchid species are deceptive, and we're looking at around 30,000 species of orchids.
Wow.
So when we met at ESEP a couple of years ago, then you gave a very awesome and very hilarious talk about these sexually deceptive orchids. That was your PhD research. Can you go into some detail on that?
Yeah, that's the system I've been working on for the last 5 years, and Australia has many, many species of sexually deceptive orchids, and it looks like we've got several different independent evolutions of sexual deception. But basically, the idea is that the orchid emits a floral volatile, which is, it can be the precise same chemical compound of the sex pheromone that the female wasp emits, and it's usually wasps in Australia at least. And then the males, unable to tell the difference between the flower and the female, come down and attempt to mate with the flower. And then they have pollen placed on their back at that point. And then by making the same mistake again, they affect pollination. Yeah, it's an amazing system. And often the morphology of the orchids is reduced to black, shiny, and warty surfaces, so they actually start to resemble insects a lot of the time.
Cool. I think you were saying as well that it was to the point where the wasps were ejaculating into the orchids as well, or did I just make that up?
No, there is one species, Anne Gaskett was the author. She did a PhD on one species here in Eastern Australia called, the genus is Cryptostylus, and they attract an incumonid wasp and excite it to such a point that it actually ejaculates on the flower. And she did that. I think she might have used UV to detect the wasp's ejaculate on the flower. So there's a little bit of one of those, hotel room cop techniques.
So what I'm hearing
on the ceiling.
What I'm hearing is that when you're in Australia, you do not stop and smell the roses. It's it's not going to be what you're expecting.
Yeah, you never know what's been done to it before.
little pollen. Oh, that's not pollen. So that brings us back to Australia. So you said you were doing a different project in Australia where you're working on root fungi in association with orchids. So you're doing all sorts of stuff revolving around orchids and both the top ends and the bottom ends, it sounds like.
Yeah, I said I would never become an orchid person when I started the PhD, but it seems like I've just continued to find projects on orchids. But the project in Western Australia, we're looking at a very diverse group of they're called spider orchids. The genus is Caladinia, and they have sexually deceptive species and food deceptive species, and they're a massively diverse genus. And we're looking. I have a colleague who's doing a lot of the pollinator work, and I'm looking at patterns of specificity in root fungus, and just to find out the extent to which that might control distributions or drive diversity in these things. Because we're still explaining why we have so many species of these orchids. Yeah, and it has a bit of a conservation spin-off too, because we do have numerous endangered species in that genus, and I'm looking at techniques that might be able to be used in order to assess soil at a site for its suitability for establishing shade house grown orchids to supplement populations and that sort of thing. So yeah, it's nice to be doing something that I can immediately point to an application to too.
Not just theoretical in your regard.
That's right, not just noodling about in the field with fake flowers and long-tongued flies.
I don't like any of this applied shit.
Awesome.
Yeah, I gotta wonder. I mean, I'm probably too focused on this because I'm stuck in the middle of grant writing. But how do you write grants and say, well, we're gonna cure cancer using orchids?
Yeah, I've been lucky to have a good supervisor who's he's developed a research program that's pretty cutting edge in orchids now, and I mean he's at the stage where the research group and collaborators are now looking for the specific genes involved in expression and emission of mimicked sex pheromones, and when we find that, we'll be able to develop some pretty strong ideas about how speciation is progressing in these very diverse groups. So we're able to just build it on a fundamental science level a lot of the time, because we're right at that cutting edge of finding out some of the mechanisms of diversification. So it's been nice to be on a project that we don't necessarily have to continually bend towards cancer or climate change. I did read an interesting review of one of his grants though recently, which suggested that one of the genes that's involved in synthesis of the mimic sex pheromones is in the fatty acid biosynthesis pathway. And so one potential spin-off by a grant reviewer, an anonymous grant reviewer, was that perhaps finding the underlying genes to sex deception could result in treatments for obesity, because it was in the fatty acid biosynthesis pathway. I think that's drawing a very long bow, but anyway.
Oh, that's weird.
Yeah.
And I'm glad you get to do fundamental research. I'm getting so sick of this applied focus bullshit.
Yeah.
I hope you're leaving soon, Stephen, because...
Oh no, I mean, I've gotten away with it too here, and it's great. But everywhere I turn, it seems like there's another article or another focus on how are you going to apply that? I don't know.
It was very liberating to see in South Africa that they still have a system that's set up to reward fundamental research. Because it rewards all research outputs, you essentially get an incentive of grant money every time you publish a paper. So labs that publish a hell of a lot of papers get a lot of discretionary research funds, basically, and they can build on that. And I think grant proposals might have to have some sort of applied context. However, the money that you get through the incentive scheme can be funneled into whatever you like. And my supervisor's at such a level now where he can fund the research that he wants to do, regardless of that climate. So it was a really refreshing environment to be in. That's for sure.
Just a technical question: is that tied to like the impact factor of the journal or something? I mean,
no.
Do you get more money if you publish in Nature?
No. It's just a blanket, incentive per publication. So.
Do you get it taken away if it's retracted? I was thinking this because you said, do you get more if it's a higher impact? And I was like, well, those papers get retracted.
I'd be surprised if the administration could get itself together to such an extent to retract money. I mean, I think people are having a hard enough time getting them organised to be paid over there.
Yeah, I don't like the effect that it sort of trickles down. At the funding level, we have to, hand wave how this works for cancer people and say all this crap when we're already, devoting our lives basically. Like, right? We've taken a vow of poverty. We're hardworking. I mean, it is it is a privilege to do what we do. But at the end of the day, it comes down to your neighbor across the hall is like, what do you work on? How does that affect us? Do you know what Parkinson's is? I'm like, no. I mean, I know what it is. That's not what I saw this article about cancer. Do you know? Is this what you work on? No, it's not at all. And I don't feel like I should have to pander to everyone. Haven't I done enough?
And I
try working on viral evolution for a while.
Yeah.
I think there's not enough of a ideological recognition that fundamental research is something worthwhile. I mean, another example from a grant review that I read was that, and it comes back to Stephen's point about how you justify orchids. The reviewer of the grant actually said, in one of the fields that asked for the economic and national benefit of the research, he said, what was the economic and national benefit of Picasso? And I think that's a good point. Like most members of the public can acknowledge the value of the arts without having an immediate applied context, and I think science can straddle that spectrum from being an economic engine in terms of innovation, all the way through to just being a generator of what's the word? or just something for humanity. It's a fundamental thing we do is ask why.
Yeah.
That's a classic volunteer's dilemma, though. I mean, everybody will say, oh, the arts are great, but nobody actually wants to pay for them.
Yeah.
Right.
Yeah, I think if we all banded together and people, when the public asks us what we do and why, be like, if I'm honest, because I think this stuff is cool, and I've gone to school for a long time, and I'll do what I want. Like, give me some money. Give me some money.
Pretty soon we're all going to be just reduced to putting on ski masks and heading out into the street to fund our research.
If a couple people did that, I'm not saying me, but I'm saying a couple people, if they did that, maybe governments will start changing their mind, Like, we have the intellectual power to make some changes. Maybe cut this out of the edits when you do it.
Yeah, we're already putting this out.
I'm coming for you!
This is terrible.
Sorry.
Academic terrorism via Heidi. So.
Watch out.
Yeah. Anyways, so you were talking about how the funding situation is kind of different in South Africa. What's it like just doing research, fieldwork in South Africa? We tried to get you onto the podcast several months ago while you were there, and we ran into a few problems with internet connectivity. But how does that sort of stuff impact, day-to-day research?
In the field it's fine, but I did manage to get locked out of the building on average once every 10 days. Because my swipe card would just stop working. I think there are some administrative and infrastructure inefficiencies and challenges over there that you don't face in a lot of other parts of the world. And I think if you go in there accepting that, then it's fine. And as long as you're surrounded by a good lab, which I was, then I was happy to deal with it. And in the field, when you're on your own with just the field and the flies and the flowers, then it's fine. You don't notice any of that stuff.
So the other thing, you're quite an accomplished photographer, and you take some amazing photos of some of your field sites and field organisms. We'll have to put a link up to your Flickr site in particular on the blog. Do you find that taking those photos kind of helps convey your research, and especially because it's not overly applicable, do you think that brings some of the research to life for these people when you're trying to explain it?
Yeah, I think so. I hadn't really thought about that so deeply, but I think people definitely engage with my talks more when you've got a picture you can put up and it makes people go wow. Then it does help to stand up there and deliver the content when I can back it up with those images. Because I do get a lot of people who tell me after talks how much they enjoyed it, and then they'll mention the photographs. And and I had a talk recently at Questacon, which was about photography and science photography, and I just went through a lot of my photos. And the other thing too is if you need to fill in if you need to fill in a minute in a talk, then you can just pop in a little slideshow as well. So
it's a useful skill to have.
That's right. Yeah, I hadn't thought of that, but I think it's definitely true.
Do you use a lot of your own photographs in your papers?
Yeah, I have used a few of my photographs in papers.
Are you always submitting for the cover?
Yeah. I always try to get an image for the cover, but I haven't managed to achieve that yet. But we'll see. I've still got papers from the PhD coming out, so I'm hoping I can get one up for that. Yeah, it's a bit of a trade-off in the field though, because the best weather for fieldwork is also the best weather for photography if you're talking about shooting plants and insects. And so you have to choose whether you're going to collect data or whether you're going to take photos, and most of the time you should probably be collecting data. So sometimes it's nice to be able to just put down the pad and the pen and the field gear and spend the time with your subject taking photos rather than collecting data. Sometimes it's a really good way of mixing a hobby with a job.
So have you still got your little boxer mantis that you had the video of on?
No,
It was on Jerry Coyne's site, wasn't it?
Yeah, that's right. He picked that up. I was pretty happy about that.
Yeah, that was cool.
She died just before I left South Africa, which is probably good timing, actually. Because otherwise I would have had to foist her on one of my colleagues and say, here's a mantis. Now you are her guardian.
And she gone through all of her molts.
Yes, yeah. I found her as an adult. And
Okay.
she was an amazing little pet. It was just completely tolerant of being handled, and you could put her on something for a photo and she'd sit there for as long as you needed her to, and she'd feed almost out of your hand. You release a fly in front of her and she was straight on it. Yeah, it's a really cool little insect. And now she's pinned, actually, so I should give her to a museum or something.
She's part of science forever now.
Yeah.
Her legacy lives on.
She's probably an undescribed species, I don't know.
It's possible. Very possible. She could have a very important place in mantid taxonomy history. So
I hope so.
Yeah, I think they make really nice pets. I bought a devil's head flower. Is that what it's called? You know what it is? A devil's flower mantis? Those massive ones that put their arms up and have all the colors?
Yeah.
I bought one of those for my man for, I think, his birthday. But it came really small and it just had a really bad molt and died. It was horrible. But yeah, you pinned her. She was amazing, though. She was just absolutely beautiful. Like when you would feed her and she was like, would display. It was amazing. But I think she went through like her 5th molt. And then, man, I mean, there's a reason where they have like hundreds of babies. Because I don't know how that's not an evolutionary stable strategy at all, right? Like they bump anything, it broke my heart. She was so pretty. But yeah, she was in the freezer and then he pinned her. Because that's the ultimate sign of love for an entomologist, isn't it?
Yeah, I think so.
I think I'm just going to cut that clip out. She was in the freezer and then he pinned her. And just loop it on something.
Yeah. I wonder sometimes if a cat dies, if, I'll come home, she'll be staked down or something. It's a horrible thought.
I was just pinning old stick insect pets last night, actually. So it's quite a good conversation to have. And I was picking up one of them to put it into a framed shadow box, and I had it on the pin. And this was the first stick insect that I'd had of this species, and I'd collected it from the wild, and she'd lived a long and full life. And then I had her on the pin, and I noticed there was a bit of a cobweb, and I went to blow it off, and then I just dropped the pin, and then I dropped her, and then snapped her legs off, and her abdomen came off. And
It's horrible.
We then conducted surgery on her with glues and pieced her back together. Put her back in the frame, and you can't tell.
Yeah. I think a stick insect, like get a parthenogenetic pet. I mean, that stays forever, right? Your kids can't mess that up. I think that's sea monkeys, pet rock. This is way better.
It is, and then it creates a tangible lineage all the way back through the family. Every family should pass a stick insect pet down the line.
Yeah. It's true. That's the best heirloom you can have.
It is.
Okay. So now that we're on heirloom stick insects, I think we'll wrap it up there and just let people consider that thought and run out to their pet store. So I want to thank you very much, Michael, for coming on and indulging us with your research. And where can people follow along as you're traveling around the world looking at orchids and cool flies?
Probably Twitter is the easiest place to go to. Mikey_Whitehead.
Awesome. We'll link all these places out for you as well on the Breaking Bio blog, which is at breakingbio.com. So thanks again, Michael. And if you want to follow us on Twitter, you can follow us at Breaking Bio. We'll see you next week with an all-new exciting guest.
Bye.
Thanks,